So first I would like to give you a little bit of an intro of where I work, because I usually do this in science conferences also, because most scientists are not used to this idea of the fusion between arts and sciences. And actually I do work in an institute that belongs to a foundation that does care about this concept of merging the arts and sciences. So as you can see here, the foundation has two museums, a museum of modern art and a museum of non-modern art, so of classical art that includes Greek statues as well as Rembrandts and a number of other classical art. We also have an orchestra, because music is also one of the arts, and we used to have a ballet. In practice, we still have the ballet. So this is the Fundação Carlos Gulbenkian, and this is also another unpronounceable phonetics for non-Portuguese. So this is very difficult to say. It's typical to Portuguese language. So I'm going to talk about body shape and the science behind body shape. And hopefully by the end of the talk, you're going to remember three words. The first word is light, which connects to the previous talk by James. And then I'm going to talk about body, and I'm also going to talk about neurons. And particularly body shape, when you think about body shape, at least in modern culture, you think about obesity. And obesity has been noted by a variety of artists across the centuries. So here we have a very recent example of Botero's Mona Lisa, in which the word obesity pops into your mind. This is not something that it's typical to the modern era, so this is 1977. This was also noted by older artists, and here we see an illustration of a fictitious character named Pantagruel. Pantagruel was a character of a novel by François Rabelais from the 1500s. And this character had a very peculiar behavior that probably mimicked the behavior of real people in that society. So the behavior was that this man had a voracious appetite. He was giant and he was extremely obese. If you move back some millennia, you also see another sign of artistic expression which was deeply inspired by obesity. Here we're looking at the Venus of Willendorf, which was not found in the United States, it was found in Austria before agriculture was invented. And of course, we don't know the artist that sculptured these sculptures, but there's a variety of sculptures like this one spread all over the world. So obesity has been around for a long, long time, but it's not until recently that it has been regarded as a disease and something that is worrying the public health authorities. So scientists started working on obesity not so long ago. And when I mean working on obesity, I mean by trying to understand obesity from a molecular perspective. So trying to understand what are the biological components that underlie this disease. And this has happened not so long ago. So here we see 1995 was when this hormone, so leptin is the name of a hormone that explains this body shape. We're looking at the child from Turkey, and this child was morbidly obese. And when I say morbidly, it means that this child had diabetes, this child had hypertension, this child had high cholesterol. So this child had serious health problems. And he had a voracious appetite, just like Pantagruel. You basically had to lock up the fridge, because this child would like all kinds of food. And even when this child was hospitalized, this child liked hospital food, which is really, really unusual. So this child had a problem, and the health problem was identified as being a genetic mutation, which was running in a family that affected only one quarter of the members of that family. And the genetic mutation was a mutation in the gene that encodes for this hormone. If you inject this child with the gene product that it's missing, you normalize their body shape. You normalize their behavior. Basically, this is a normal child that has been cured by injections with the hormone that it's missing, and this is a child before, the same child before that injections. And the wonderful thing about science is that it doesn't work just for humans. It also works for any of the mammals that bear the same mutation. So here we have the same disease in an animal model. And actually, this is the animal model that enabled that child to be cured. This is where leptin was found. So leptin was found by Jeffrey Friedman, with whom I have the pleasure to work for 7 years. And Jeff mapped the mutation onto the chromosome 16. So these are the saviors of the child. These are the guys that enabled us to be able to cure a certain subtype of inherently genetically encoded obesity. So what does leptin do? Leptin, as I mentioned before, is a hormone, and you can call it an adipostat hormone in the same way that you would call a thermostat, a machine that controls the temperature to keep it in a very narrow range of variation. So this hormone is produced by the adipose tissue, and this is a very reductionist form, or the reductionist representation of the adipose tissue, which is basically a collection of fat cells that look like ping pong balls. We'll get into that. So the adipose tissue produces the hormone leptin in amounts that are proportional to the amount of fat. So the more fat you have, the more leptin you have. And the more leptin you have, the more it's going to signal in the brain to tell you stop eating. So that will decrease the food intake, which in turn is going to reduce the amount of fat, and thus the amount of hormone that the fat produces. So this is a typical negative feedback loop, just like a thermostat, so it's an adipostat, that keeps the amount of fat in a very narrow range of variation. So as the fat diminishes, the amount of leptin is also going to diminish, which in turn increases the amount of food take, which then in turn makes the fat go back to the original size. So a lot of biological processes are based on these principles, that there's a biological process where you want to keep boundaries and you don't want to have it shoot off size. So these negative feedback loops, it's a common theme in a lot of biological processes, like ovulation, like thirst, like many other physiological processes. And it's not until recently that finally we got a hold into the machinery that controls adiposity. And this was only achieved in 1994. So it's very, very recent. Ever since then, we've been trying to map out the neuronal circuitry in the brain, but also outside the brain, that controls or that mediates this negative feedback loop. And we have been doing this thanks to light. So this makes the connection with the previous talk by James. So here we're looking at optical fibers that conduct light, and the technique that enables us to do the science underlying obesity, it's the technique named optogenetics. So as the name indicates, there's a component that involves light, so that's the opto, and there's a component that involves genetics. I'll get into that. So this technique has been popularized by Carl Deiser-Roth at Stanford, with whom I had the pleasure to work for a few weeks. And I got trained in optogenetics, and as you can see, the light fiber is doing something here, and it's being implanted into the brain. So I have to make a clause here that none of these procedures produce pain, because actually your brain is painless. The brain doesn't have any pain receptors. And all of the procedures that have been applied to animals. They have been extensively approved and scrutinized by ethical committees that usually involved members of society. So as you can see here, the optical fiber is being plugged onto the brain of an animal. And why is that? Why would the light affect the brain? The light affects the brain because we're taking advantage of genetics and transgenesis. So this is now the part of genetics that I will explain. So optogenetics relies on a channel. So this is basically a protein that is sitting in the membrane of deep sea algae. So these creatures, they're unicellular. You cannot see them with naked eye. You have to use a microscope to see these creatures. And they have a flagellum, just like sperms. So basically, they're the same size as sperm cells. And they use this channel, which is sitting in their membrane, to move the flagella. And why do they have that? So nature developed this light-sensitive channel. So this channel is usually closed. It doesn't let permeate anything. But in the presence of blue light, the channel opens. And why blue light? These creatures, they live deep down in the ocean. And deep down in the ocean, the only wave light that penetrates is basically blue light. And below that, there's nothing. It's darkness. So these creatures, they use this channel in the presence of blue light in the ocean, deep down in the ocean, to move the flagella towards the light. And this is called phototaxis. If you use this channel in a different context, then you're going to enable that context to be sensitive to light. And this is what neuroscience has done. We have been able, using transgenic tools, to deliver a gene from a different animal into the mouse brain. So here we're looking at neurons in the mouse brain that have been transformed so that they now produce this algae channel rhodopsin. That's the name of the gene. And by doing so, by producing this channel, we can drive the neurons to become active with a flash of light. So here we're looking at the raster. This is basically the response of a neuron. And then whenever you flash the light, you see that the neuron, boop, 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 boop. This is the activity of the neuron that you can record with an electrode. And here you can turn on the neurons on and off at will. So here we have a 5 hertz flashing of the light. Here we have 20 hertz. So basically, we can control the neural activation with channel rhodopsin by using light. And with this, as we've shown before in the first picture of a rat, we can implant these optical fibers deep down in the brain to stimulate whatever neurons you want that are affected by channel rhodopsin. As you can see here, so this is my data, and the previous data is also my data. We're able to implant these fibers, and here, this is an MRI image of a brain, of a mouse. You can see the fiber track, which is pointing down to an area in the brain, and using MRI, we can see that the flashes of light, with the flashes of light, we have correspondent neural activity. So this is the ups and downs of neural activity that we can promote using flashes of light. And why are we doing this? We want to do this because we want to know what are the neurons that are controlling behavior. Particularly, we want to know what are the neurons that are controlling appetite. And here we see an example. This is not my data. This is data that was produced by Jessica Aponte, which now works at NIH. And she was a postdoc with Scott Sternson at Chilean Farms, and she produced this data together with my collaborator and colleague, Denis Atasoy, who now works in Istanbul. So here we're looking at the mouse. The mouse is basically resting. It's sleeping. And you can see here that the light is starting to be flashing. It's starting to flash. You see the light? Okay, so that's the light. And soon after, the mouse is like, oh, what's going on, man? And so here, there's a dispenser with food pellets, so little balls of food, and the mouse starts eating. So basically, in the middle of the night, the mouse has a binge episode because the neurons in the brain, deep down in the hypothalamus, are being activated by flashes of light. And it keeps going. So basically, This is some of us waking up at night, going to the fridge, and having a binge episode. So this is one of the applications of optogenetics that illuminated the neural circuitry that it's controlling something that you actually think is under your control. This is what most people think. Most people think that your appetite and your hunger levels, they are controlled by your voluntary behavior. Where actually here, the neurons that you're stimulating, they are very far away in the neural circuitry from your voluntary behavior. So the hypothalamus is very deep down in the brain. It's an area in the brain that is common to reptiles, for instance. And the hypothalamus is not an area in the brain that you can manipulate with your willpower. So this is an example. The other example that I've used optogenetics for was to answer another question that we also think that it's under our willpower. And the question is, what mediates your likeness for sugar? Why do we like sugar? Essentially, that's the question. And this is a question that I think it's pertinent when you look at market data. So here we're looking at the market share for beverages that have regular sugar and beverages that have artificial sweeteners. And as you can see here, ever since artificial sweeteners were introduced in the market in 1947 in the form of sweetened law, that artificial sweeteners, they're not really liked by the population as a whole. So here we're looking at the behavior, the market behavior of millions of people. So this is not just your individual preferences for artificial sweeteners versus natural sweeteners. We're looking at data that is very robust because it reflects the behavior of millions of people. And as you can see here, people don't like artificial sweeteners. And why is that? I performed a series of studies that were published not so long ago, in 2011, between 2011 and 2013. And those studies identify the neurons in the brain, again, in the hypothalamus, that control this behavior. So basically what I did was to, so now imagine that this is a mouse, and that I implant an optical fiber deep down in the hypothalamus, and I activate these neurons here via channel rhodopsin activation. So I use light to activate these neurons, and I do this when the animal is drinking the artificial sweetener. As a consequence, I am able to invert the natural preference of the animal. Oh, am I with time? Okay. So moving on. More recently, I chopped off the brain from my interests. So here, there's no brain, but there is a huge neural network that innervates the body. And the one problem you see with this textbook image, so this is a book that medical students use in medical school to study anatomy. And of course, most textbooks, they are very incomplete, particularly if they were developed in the mid-1700s. And this is a problem with this image, is that this image doesn't have any modern knowledge. At least doesn't have any molecular knowledge for sure. And that's evidenced by the color that everything is painted yellow as if all the neurons are the same. The other problem with this image is that this atlas obliterates an organ that is very important, and that organ is an endocrine gland. And the endocrine gland that I'm talking about, it's basically the fat tissue. As I mentioned before, the fat tissue produces hormones, and any organ that produces a hormone is by definition an endocrine gland. And if you think about the fat tissue and you need to draw it, so if you're a draftsman, an anatomical draftsman, and you're trying to draw an organ, you can draw a heart. Everybody knows how the heart looks like. You can draw a pancreas. But how do you draw the fat tissue? So this is a question that we try to address using techniques that I'm not going to get into details of those techniques, but it's basically a relatively recent technique named OPT, optical projection tomography, which enables you to look through the tissues by making them transparent. So this is how the fat tissue looks like, which is very different from a collection of ping pong balls. We can see that there's a lot of detail in these images. We can see that there's actually neurons that are innervating the fat tissue. And this is a sizable sample. This is about 2 centimeters long. We can see these neurons actually with naked eye, and we can dissect them out, and we can ask questions about these neurons. So here we're looking at different stains that we performed in these neurons, and we can see what's the molecular identity of the neurons in the fat. We can actually put an animal under the microscope so that we look at these neurons in vivo in a living animal. And we can, this is one such image, and we can trace down those neurons and see where do they end. And then for the first time we saw that actually neurons, they can project onto an adipocyte. So we have this neuroadipose crosstalk between neurons and the adipocytes. And if you have that, what's the function of that? So here again, as a scientist, you want to know why things are there, and how do they work, and can you tweak them to work more? And this is what we can do with channelrhodopsin. So again, I am able to put channelrhodopsin using transgenic tools. I am able to produce channelrhodopsin only in these neurons. And then we activate the light by implanting optical fibers subdermally, and we can ask what happens to the fat. So if we prescribe light therapy to these mice, and we measure the amount of fat with MRI, so here we're looking at the sagittal section of a mouse, and we can quantitate non-invasively the amount of fat that this animal has under the skin. And we see that with light therapy, animals that do not have the deep sea algae gene, the channelrhodopsin, so animals that do not have the channelrhodopsin, they're not sensitive to the light. And if they're not sensitive to the light, then you prescribe them light therapy only in one, basically on this side, but not on the other side. If you prescribe light therapy on the right fat pad, nothing happens. So basically the right fat pad equals to the left fat pad here in yellow. But animals that do have channelrhodopsin in these neurons, you see that the fat pad on the illuminated side disappears. And it disappears because the fat pad and the adipocytes, they engage fat breakdown through lipolysis. So bottom line, we discovered neurons that can control body shape. So these neurons, they can control the amount of fat in this mouse, only on the right side. But this is not to say that now we want to use this knowledge to create a reverse body shape, right? So this is reverse body shape. This doesn't exist in the human population. Actually, if you make a Barbie that actually has the average morphometric features of a normal woman, this is how a Barbie would look like, right? So this doesn't exist. And our society is a little bit obsessed with changing the body, pretty much in ways that have been documented before. So these are the various ways that people have been torturing their body. Any questions? Okay, so this is also torture, or this is how we classify torture. And this is no other ways of creating torture. Probably, if you use anesthesia, you don't feel the conscious perception of pain, but there's many other different levels of pain that can be psychological or psychiatric that come from trying to move this into that. And there's, of course, a whole industry that feeds on this notion. And, of course, this is all fake. This is not science, basically. So I'm a scientist, and I know that none of these pills has a mechanism of action that actually is based on any scientific proof. And this is not just the nutraceuticals, as these are called, because they're not pharmaceuticals. They're not based on mechanisms of action. There's also another type of industry that it's feeding on this notion that you can actually change body shape. And this is kind of a headache. If you try to understand what these food packages tell you, I don't know, is there anyone that actually knows how to read these things? And you look at it and you're just like, oh, fuck it, I just go ahead and I do. And I buy whatever. So if you look at these food packages, I mean, here you look at the various nutrients in these packages. Here, for instance, you see that there's the dietary fiber. So this package, it's really confusing, right? Because they don't put this in a clear way. You have to do a lot of back of the envelope calculation just so that you get to an amount of how much fiber should I eat today. And if you do the calculations, this amount is about 20 grams of fiber, which is basically a lettuce leaf. Really? I mean, really? Do I have to eat one lettuce leaf a day to get fiber? That's easy. Obviously, there's a lot of things here that I quite don't believe in it. And also about calories. Why do they put this amount of calorie here? Who in the audience knows what's a calorie? Raise arms. What is it? You know, you're a scientist. 2, 3.
Well, I think I know.
Okay, let me give an introduction about what's a calorie. So calorie is a very old notion. So there's nothing modern about that food labeling. It was introduced in 1782 by Laplace and Lavoisier, who were scientists a little bit like we are these days, that you know a little bit of everything. They did biology, they did math, they did medicine, they did a bunch of stuff at the same time. So those were the days. And basically they developed this, and they were also engineers because they developed machines. So they developed this machine, which is basically a chamber inside a chamber inside a chamber. James, you were talking about chambers. These are chambers. So this is a chamber in which on the outer part you put ice. And you can see here that the ice chamber is connected to a little tap. So it's connected to a little tap, which is this tap here. And on the inner chamber, so this is a basket where you put food, where you put whatever. And you set it on fire, you close the lid, and while there's oxygen in the chamber, the food is going to burn. And when the food burns, it creates heat, and the heat melts the ice. So basically, in the 1700s, calories were measured with milliliters of water that would drip down from this tap. Okay, so calories is basically heat that is produced through combustion. So we all know, and this is what nutritionists tells us, and there's a lot of nutritionists out there, that fibers do not have any calories, right? So that's why you have to eat all veggies. Veggies have fiber. And actually, this is fiber. Fiber must have a lot of calories, right? Because fiber is cellulose, cellulose burns. So actually, I really don't buy this number. Because if anything, something that has a lot of fiber should burn a lot, therefore should have a lot of calories. So from a cow's perspective, you see that the cow doesn't really know what to read. Am I going to look at the fibers content, or am I going to look at the calorie content? Because cows, unlike us, they can digest they can digest cellulose, and they can digest cellulose because in the gut microbiota, they have enzymes that can convert cellulose into glucose, which is the molecule that our brain can eat. So I don't really know what to think about these food labels because they really don't disclose all the methods that are involved in coming up with these numbers. My gut feeling, and using the reasoning that I just described, is that these numbers are not accurate because fiber should have a lot of calories, just like alcohol, just like gasoline. And if people that do these food labels, if they cannot explain it to me in a simple way, maybe it's because they actually don't understand it very well. And if you don't understand something well, there's another artist. There's an artist that actually said very wise things, that if you believe in things that you don't understand, that's superstition. Yeah. So science is not about superstition. Science is about creating a body of work that is validated and scrutinized by scientists that will corner you if your methods are not clear, if your methods are somewhat not very transparent. And that's why all of these journals, I think they produce knowledge that I can believe in, because if I dig down into the methods, I'm going to be able to understand what's written there. And there's different levels of journals that one can read, from very specialized journals all the way down to journals that convey information to a lay audience. And unlike superstition, science, it's the hope that patients can have. So this is just a movie from a living adipose tissue where you can see that there's a lot of complexity there, and of course, I'm trying to understand this complexity, me and a lot of other people. This is more images that I find them to be aesthetically pleasing. They don't have much concepts behind it because we still don't understand what we're seeing, but we'll get there. This is my lab. So my lab is very gender balanced. And I have to thank, Andrea was not there at the time we took the group picture. She is also someone that works in my lab. I have to thank to a lot of the funding agencies that are helping me to create science. And of course, I have to thank the Fundação Calouste Gulbenkian and the MIT. So Roxana is an MIT student. And the Human Frontier Science Program that just gave me €1 million to perform more research. And EMBU and the National Funding for Science Foundation. And the Gulbenkian Foundation also has art. Oh, where's Camille? I don't know what happened to this picture. So we have a musician in residency. Our institute, from the Gulbenkian Institute for Science, has had 3 musicians in 3 artists in residency. This is a new program that the institute is implementing. Marta Menezes and Camille Van Nuenen, who is a composer, and Rob Kessler. So they have been very nice people to have around, and certainly they breed well with us in terms of conceptual production. And I'm organizing a meeting that is also disappeared. Well, I don't know what's going on with this. I see the images here, but it's not. Okay, any questions?
Can I use your computer?
Yeah.
So, thank you. That was interesting to update a little bit on the fatty tissue. So I was intrigued by the picture you showed, the neuronal innervation of fat cells. It looked amazingly precise. It seemed that it was just one cell that was innervated. So why would we have such a precise innervation, and why neuronal? If you want to burn fat tissue, there are other ways to turn that metabolism on. Why would you? It seems like it's too precise for the purpose, right? You want to use nerve signals to control muscles where you need movement. So are you sure you are not stimulating muscle? I mean, there are always muscles, smooth muscles, and striate muscles close to these fatty tissues. I mean, I thought about the rat experiments. I mean, if you would locally contract muscles, would consume energy, and maybe locally derive energy. So that's. Do you find it surprising that it seems so very precise?
One of my reviewers didn't think it was very surprising.
Yeah.
Different brains think different things. So precision is.
Other reviewers thought it was. But the other reviewers thought it was surprising. Or
we had 2 reviewers that felt that this was knowledge that had to be. That validates a point. It is very precise, and actually biological systems, they can have both redundant mechanisms, but they also can be very precise. And of course, evolution played a big role in selecting for things that are actually precise, because whatever it's not precise usually gets filtered out.
I disagree with that. That's,
okay, so you disagree.
Yeah.
But those are sympathetic neurons. They're not neurons that innervate skeletal muscle. And this is defined by transgenics. So it's the molecular identity of those neurons that we validate molecularly that those are not muscles that innervate the skeletal muscles. And if you think of what are the ways, I'm open to discussion. You tell me what are the other ways.
Okay, so one more quick question from Marcus.
Congratulations to this really fascinating
Thank you.
new concept. A very simple question I have. When the fat tissue gets more and more, and there are more and more cells. Do you insinuate that there will be more neurons?
Not more, less.
Less? So it's not a cell-by-cell contact.
So the connections are very sparse. And actually, there will be a paper that will come out in Cell soon that demonstrates that you don't need a lot to make a lot, because the fat tissue has an amplification system based on gap junctions. So that one unit, one adipocyte, is able to communicate with the surrounding adipocytes to create a wave of beiging. Now, beiging sounds like a color, and it is a color. So there's two types of adipose cells. There's the white adipose cell, which is considered to be the mean cell, and there's the brown adipose cell, which is a fat cell that is specialized in producing heat. And heat is a way of dissipation of energy, of biological energy. In between these two colors, white and brown, you have beige, which is another cell type, that it's an intermediate type of cell between the brown and the white. So this intermediate beige cell also produces heat, not to the same extent as the specialized brown cell, but it is the heat-producing cell that humans do have.
Thank you.
You're welcome.